talking about how you quantify fluidity. And there's two types of data that you get from this measurement. The first is the diffusion coefficient. And what that is just the first derivative of that line. It's just the slope of how fast that goes up. So the faster that curve goes up, the faster the protein is diffusing. You can actually extract the diffusion coefficient. Now, the diffusion coefficient is inversely related, so it's 1 over the square root of the molecular weight. So the bigger the protein, the slower, because that just makes sense, doesn't it? So a protein that was smaller than this one, had a lower molecular weight, and had the same type of mobility, would actually diffuse faster, so the curve would come up and be higher. Now, so that the speed of recovery tells you the diffusion coefficient in that particular feed. And the second piece of data you get is, to what extent is that protein confined? Is it bound to something, or is it mobile? And so, if this line goes all the way back up to the starting point, then you get 100% recovery. So the recovery rate tells you how mobile the protein is, whether it's attached or not attached, free to move or not free to move. So what happens is it goes up to 80% of the original value. What that means is only 80% of the proteins are free to move. 20% are actually bound to something and confined, right? And if you have a protein like she showed, so the other thing you can do is you're not restricted to looking at proteins on the plasma membrane, because with confocal microscopy, you actually take an optical section anywhere through the cell you want, so you can zoom down and look at the nuclear membrane, which is what she was looking at. And that protein that they were viewing had no mobility, because it was attached to what? The nuclear lamina. And so that protein's not going anywhere. You get zero recovery. So this line would have no slope, and it would have no recovery. It would be 0% recovery for a protein that can't go. Right? So this, you get a couple of different kinds of data from this measure. Okay, any questions about that technique? We're going to do a problem with it here in a few minutes to see if you can understand. All right, so this is the whole issue of what are the possibilities? You've got, so that's the problem with that technique is you're doing a population, you're looking at the population of proteins. You're not looking at one protein. So it's the average diffusion coefficient. It's the average recovery, right? But we now are able to, we can attach beads to single proteins. Okay? And we can view those beads, so it's called single particle tracking. And so we can actually look at the movement of individual proteins. And so you get different patterns according to how mobile that protein is. And so if this thing can just zigzag all over the surface of the cell, that means it's not restricted to any particular domain. It's completely free to recover, so it has complete mobility. If you can have a protein like this one, this is a red blood cell, and it sort of stays in one area. There's actually a better slide than that. Yeah, so it starts out here, and it kind of stays in this domain for a while, and then it comes over here, and it's corralled in here, and then it stays there. So it's having discrete areas where it's bumping around, and the reason for that is the nature of the red blood cell. So we talked about the red blood cell as a good example of direct and indirect peripheral membrane protein. Okay? But you can also look at the proteins that are transmembrane. Okay? And so this is a little corral that's formed by the proteins, the cytoskeleton that's underneath the plasma membrane. And so what kind of protein is going to interact with that? Well, peripheral membrane proteins can interact. So if a protein is bound to this, a protein like anglin, if you measured its frac, what would the frac look like? None. It has no slope. It's completely bound to that membrane. But there are proteins like this one, span free. Some of it you can see is attached, so some fraction of it is going to be immobile. But some fraction is going to be corralled. And so if you put a single particle tracking device on that beam on top of this guy, what you're going to see is it's going to be bumping around inside the corral, and then it'll break through there because that's what happens. The proteins stay together only for a few milliseconds, and it'll leave, they'll get caught in the next corral. That's what was happening in that previous diagram where you have the thing is corralled and it moves into the next corral. So there's places where you can trap it. And so single particle trapping gives you a really good estimate of what's going on underneath the cell membrane and how it's being restricted. So if you did a frap of band 3, would the curve go all the way back up, or would it go part of the way? Just part of the way. How high it goes, like it's 50% or 20% or 80%, is the percent of protein that's free. The difference between that and the top, 100% recovery, is the fraction of decimal. So if it goes up to 80%, it means 80% of the proteins are in this state right here. It's confined, but they're mobile, right? And it means that 20% are actually bound to the cytoskeleton and can't go in. So this is a really useful technique, single particle tracking. And so you can get several different possibilities. You can put a bead on a protein, and sometimes they go like this. They go in directed motion from one end of the cell to the other. That's not random. So what do you think is happening with that? Well, it's on a microtubule. So it's connected to what kind of protein? The little guy carrying the vesicle. It's a motor protein. Yeah. So when you see that kind of behavior, you know that thing is being bound to a motor protein that's walking on some cytoskeletal element, namely microtubules or actin filaments. Okay, so you have different things, you get different patterns depending on what's going on with the proteins that they're interacting with. So here's a couple of ways that you actually get aggregation. Yes.
Real quick.
Zero. What it comes up to is the percentage. If it's 100%, it's 100% mobile. Yeah, unattached. If it's 80%, so it's the height of that curve that it reaches. What fraction of the original fluorescence does it achieve? And that's the fraction that's mobile. If it's 20%, it kind of just comes up a little bit. It means only 20% is free to move around, and 80%. So the remainder is the fraction that's bound to something. If it's 100% bound to something, it means that we know that it'll just be a flat number. Okay, so here's something that would cause 4 different scenarios that cause confinement of protein. And so this is what happens with bacterial rhodopsin, that weird purple bacteria. And so it's actually called that because it's got purple patches, and that purple color comes from the retinal pigment in the bacterial rhodopsin multipass protein. That retinal pigment is what's absorbing the light, and the protein is using that absorption of light to pump protons out of this part of the membrane. But they're almost like little photoelectric cells. They aggregate together, as I showed you with that freeze fracture electron microscopy, and so they just form these plaques. And so they're not going anywhere. They're stuck to each other, and they're just like, it's like putting an array on top of your roof to get sunlight and generate electricity. So that's one way, self-aggregation. Number 2 is, if it's a membrane protein, it can be, and it's crossing the plasma membrane, say it's a multipass membrane protein, it can actually bind the proteins outside that is in the extracellular matrix. So the exoplasmic compartment, the extracellular compartment is out here, and that's not empty, it's full of proteins. And so if this membrane protein attaches itself to the extracellular proteins, guess what? It has no mobility, 0. The only fraction that's going to move are ones that are not bound to that extracellular matrix. And you can have extracellular proteins trapping membrane proteins. As I just showed you with the red blood cell, this is like the red blood cell. So there are cytoskeletal proteins that are grabbing on to those membrane proteins and holding them steady. So that's going to be a method of defining that. And then finally, you can have this situation where two proteins are joining hands, and when they do that, they actually form a type of tissue called an epithelial layer, like your intestinal epithelium or your kidney, renal epithelium. And epithelia form a barrier between two compartments. And to do that, they must hold hands like this, so each protein is extending the protein hand, and those two proteins are holding or bound together. And what that does is it forces nutrients and things in this compartment to go through the cell instead of go through between the cells. So, but if you did a FRAP analysis of this junctional protein, what you'll see is no movement whatsoever. Because it's actually meant to prevent things from moving between cells. Okay, so here's the example of the bacterial rhodopsin array. This is an example of, you can imagine this protein in the extracellular matrix binding to these proteins, preventing them from moving around. And then here's a good example. This is like a intestinal epithelial cell that so here's the lumen up here, okay? And these cells form a barrier to prevent all that stuff from just diffusing into your blood. You want to be very selective about what you take from the gut lumen. You want to leave the junk behind, and you want to take all the nutrients in. And to do that, you need to make the nutrients go through the cell, not between. Can anything go between them? Absolutely. Different junctions are selectively permeable to different things. Some are permeable to water. Okay? They'll let water seep through here. Some are selectively permeable to sodium. Others are selectively permeable to chlorine. So the selectivity of that junction is very precise and very specific for that particular epithelium. Okay? But the junctional proteins that are holding this together are completely immobile because those two cells are basically holding hands. Yeah. They will. And you can do that. You can actually put specific proteases there and actually, in fact, that's how we get cells to release from petri dish or cell culture. You have to trypsinize them. You don't trypsinize them to death. You put a low concentration of trypsin for a limited amount of time, and you can split them apart. It would cause what? Well, you're in control of whether you put trypsin in there, okay? So it's not an error. I mean, you should know what happens. But that's actually a good question. You know, you can say, well, if you have this epithelium, what would happen in situ, the way it operates? So you can actually take out an intestinal epithelium, you can actually evert it, you can put it between two chambers and study the movement of solutes between compartment A and compartment B. Well, if you do that, what's going to happen is all the nutrients are going to go through the cells, and nothing's going to go between them. And if you did a fraction experiment, those junctional proteins would be frozen. There would be no recovery after fluorescent bleaching. Okay? But so now, if you throw in trypsin for 5 minutes, what happens? What changes in terms of the fraction? Everybody is now mobile, including the junction protein. These guys were also mobile, but notice they're actually corralled. These sets of proteins cannot move past the tight junction. There's a reason for that. Because if you put the protein on the wrong side of the cell, you get the wrong movement, the directional movement of the protein or nutrients changes. And we're going to go through that as we talk about. Because we're starting with really simple things. Okay, we're looking at one transporter, one pump. But what's the real situation? Thousands of different kinds of pumps, thousands of different kinds of channels, all operating to do a concerted job, like move sodium and glucose from this side to that side. And so we need to get some terminology under our belt before we can handle this. Okay. Good question. Anybody else? All right. Okay, so let's stretch our muscle should this experiment. And so, let me pull up the gizmo. So answer this by yourself. Oh, okay. Yeah,
I just like it.
Okay, so it's really a discussion question. So think about what would happen, and then I'll quiz you on what yours.
Just finished for after
There's no need.
walk away.
We're going to do it. I see some people. This.
When you talked about it, I just
You were
I
correct.
walked in when you finished. So I was like,
Okay, so the way to approach this question is, first of all, both of these are close to each other, and so you have to know those two proteins. I had to tell you about those proteins in order for you to answer this question, right? So you know which proteins are there because they're close to each other. The assumption, let's make a couple of assumptions. Let's say, because you really can't tell, because you don't know the molecular weights. The molecular weight's going to affect the ability of that photo recovery, right? Let's assume that the molecular weight of those two proteins is identical. Now think about which one's going to move, which one's going to recover faster. And if you think that lactoferrin is going to recover faster, so it's going to really shoot back up, then pick A. And if you think alpha phosphatase is faster, pick B. Let's just, another assumption that's important would be, let's say they're 100% mobile, none of them are aggregated. Because you have no way of knowing, right? So I'd have to give that to you. Or I would have to show you the data, and then you could figure out whether there was mobility.
That's what they're
Couple seconds, because I want to re-answer this group. Okay, got it? Quick summary.
I will. Yeah, my nose has been all.
Okay, so, wow, this is pretty even. So, we got a lot of, not clear, right?
Someone trying
So
to
now, turn to your group, come up with a group. So, what kind of membrane protein is lactoferrin? You have the tools to say, oh, that's this type of protein, right? What type of protein? single pass integral membrane protein. That's the full description. So it has the same molecular weight as this other one, which is what type of protein is alkyl phosphate? GPI anchor. Okay. So one protein is spanning the plasma membrane. And how is it spanning it? With a single alpha helix. But that alpha helix is wading through lipid muck, right? It's in the plane of the membrane. And its diffusion coefficient is slow because it's wading through oil. Which is faster? Wading through oil or wading through water? Oil, right? Now, think about the little kid holding the balloon. The little kid's legs are the phospholipids. That's the only thing that's in the oil compartment. And that thing is zipping around, right? The balloon is the protein. So it's going to move much faster than a protein that's wading through, has its alpha helix dragging through the mud. Okay? So you've got just two little lipid legs, and they love to be in the lipid layer, and so that thing is moving rapidly. The only thing that's slowing it down is the weight of the balloon. So it should be the case that alpha phosphatase diffusion is much faster. If they're both 100% mobile, they should both recover to 100%, but the one that gets to the top fastest is alpha phosphatase. Now, what if, let's see, okay, let's focus on just alpha phosphatase and rethink about the question. What if alpha phosphatase, 20% of it is bound to the extracellular matrix? So the protein is anchored out there, so we can grab on to proteins. So 20% is bound to proteins. What's that track curve look like? Will the diffusion coefficient change? No, you're still, the ones that are free to move are still going to move at the same rate. But it won't go all the way back, it'll go up to 80%. 20% are stuck. The balloon is stuck on somebody's roof. Okay? So those are the two kinds of information you get from these track curves. It's just common sense about which one's going to go faster or go slower. Obviously, if, let's say, you have two proteins that have the same, so they're both GPI anchor proteins, and one's bigger, what's going to happen? Which one's going to move to the top fastest? The smaller. Okay, so it's just basic stuff. Now, let's do this. What if we have a membrane that has, a plasma membrane that has no lipid rash? We can actually do this experimentally. We can get rid of all the lipid rash by depleting all the cholesterol from that membrane. So you've got this guy, the alpha phosphatase. Remember, they have GPI anchor proteins have a high affinity for lipid rash. So if there's no lipid rash, you make that measurement. What's a frat curve? Okay, it's got this frat curve, whatever it is. Okay, and let's say there's no percentage is tied to any other protein. So it's free to move all over the place. And then we do the same experiment with a cell that has lipid rash. What's that curve going to look like? It won't change the level, the recovery. It'll still go back to 100%, but it should be faster or slower. Slower, because it's going to move around, okay, with two legs until he gets into a lipid raft. And then he's going to move really slowly because it's a liquid ordered state. So the lipid raft will slow the diffusion of that protein. And you can actually use that to characterize the percentage of lipid rafts in the membrane. Okay, so interesting kind of stuff. The disadvantage of FRAP, of course, it's a population study. You're not looking at one particle, you're looking at the whole group of particles. All right, so now let's see, there's actually another question. Okay. This one we delved into last time. So looking at, you should be able to look at a diagram, this was actually from the test, and figure out how that protein sits in there. And this one, let's go ahead and put that to the lead one. Okay. Let me make sure that majority of you, well, it's still just half the class. We got to do better than that. Okay. Now, turn to your group and figure out what your answer is. Oh. Wait, I'm still disturbed that only 54% of people figured out. And explain to your fellow groupers why you picked B. It's B. B is the answer. But you gotta know how to read this.
I just feel like hydrophobic. Okay. Yeah, I agree.
So, what's the first thing you know? You know where the N-terminus is, right? You know it is right here. But you don't know what side of the membrane it's on. You can't tell that. So, the N-terminus is right there, and then you can tell the length of this protein sequence, right? How many lines? And it's a little over 100, right? And this tells you what? If it's up, it's hydrophobic, right? So these are hydrophilic. So these are probably either the extracellular space or in the cytoplasm. Okay. How many transmembrane segments? You've got one peak. It needs to be about 20 amino acids long or it doesn't qualify. Okay. So there's some slop there. It's like 19 to 26. But it's pretty narrow. Okay. And it should be right in the middle. One alpha helix. Now, here's a rule that's really useful. If it's got an even number of alpha helices, membrane spanning alpha helices, that means that the N-terminus is on the opposite side of the membrane from the C-terminus. You still don't know which orientation it is, but it's got N-terminus is going to be on one side of the membrane, and this is just common sense, right? It's easy to figure that out with one alpha helix. If it's got three, it's still the same rule. It means the N-terminus is in one compartment, and the C-terminus is on the opposite compartment. Odd numbered, I say even, it's odd numbered. Means the N-terminus and C-terminus are on opposite sides of the membrane. So the corollary is even numbers of alpha helices mean they're on the same side. And you're going to see an example, hopefully if we get to that, where that's the case. So if the C-terminus or the N-terminus is, you find out that it's on the cytoplasm, that means the N-terminus of the C-terminus will also be on the cytoplasm if there's an even number of alpha helices. Now, how do you figure out which one is which? That's the next question. And we're not going to take time to actually do it. I'll show it to you. Okay. So here's the information. This is also, these were just straight from the exam. It turns out that domain number 3, right here, is heavily glycosylated. So clearly that's a hydrophilic region. Okay. And what else does that tell you? It's on the outside of the cell. So you can deduce that this domain is going to be on the opposite side of the membrane because how many alpha helices do you have? You have an odd number, 1. Okay? So that means this is cytoplasm. And that's how you can tell it. What if I said this domain has lots of disulfide bridges? Now it becomes the outside of the cell. It crosses the membrane, and then this part is in the cytoplasm. It's just real basic, straightforward analysis. Any questions about that? tests by group number. So give her a chance to do that, and then send one member of the group to come fetch your group of tests. But do not, I want the tests that if the person is not there, please return them.
Go down.
Yes. Why would you not expect to find a distinct hydrophobic peak?
Because every other amino acid inside is available in water.
Yeah, so it's beta strand is the key to answering this question, knowing the structure of the beta strand. And so what it has is every other amino acid is hydrophobic, so it would be there'd be no peak, right? The other thing is, even if it was, let's say it is a peak, okay, how would that, what would the distance of a strand required to cross the plasma membrane, how would that differ from the number of amino acids it takes to spiral across using an alpha helix? Which one's longer? Which one is going to require more amino acids, the alpha helix or the beta strand? Yeah, it's the beta strand is shorter because it's just like this. It's not wasting time going around in a circle. It's straight. Right? It's a pleated sheet, yes, but it's much shorter. Okay. So those two properties we distinguish. Okay, good. All right, so now we're into transport, and so finally we're getting to some useful functioning instead of just definition. So we do need to sort of, you're supposed to know how to categorize membrane proteins if you had Dr. Dean, but I want to remind you of the way I think it should be categorized. Dr. Artigas yesterday, that's how he started his seminar. He goes, okay, here's how membrane proteins are arranged, and here's the class, and I'm going to talk about pumps, okay? So how does it all fit together? The way to start is with the type of energy that's required to move the solute. So we start with transport. Is that it? No. Somebody tell me when I do this. Nothing's working today. Good. Transport. And so there's two basic categories. Those that are passive. That is, they're only going to, diffuse. Whatever they're going to do is going to be diffusion. Diffusion is passive. You don't say passive diffusion. Why not? That's redundant. Don't say passive diffusion. It's either simple diffusion or facilitated diffusion. But diffusion means the transported solute is moving down its electrochemical gradient. If it's not charged, it's just moving down its chemical gradient, right? But it's using its gradient to drive the movement of the solute. You can also have active transport, right? So that means you're going to put in some energy. That energy can be in the form of the sodium pump, which is splitting ATP, or you can create, using the sodium pump, you can create a sodium gradient, and then you can work transport off of the sodium gradient. You can actually move things based on the inward movement of sodium. So there's lots of different ways you can do it. So let's finish the easy one, which is passive. So this is diffusion. It's in this side, okay? And you've got a couple of different flavors. Actually, just two. You have simple diffusion, and you have facilitated. And you can categorize facilitated further by dividing it into transporters, which is the type of protein, or the channels. Those are the two choices if you're talking about facilitated. And there's another name. It's not all, there's a lot of things that are called transporters, including pumps. Okay, so you got to go below this. Uniport. if it's transporting one solute down its electrochemical gradient, or its chemical gradient, or just its electrical gradient, it's called a uniport. Now, some biochemical textbooks that don't know what they're talking about will call pumps moving a single proton by splitting ATP a uniport. That's hogwash, okay? Because there are people that do this for a living, membrane transport, people like Dr. Ortigas. The reason it's in a category like this, uniports, is because of the amino acid sequence. It's got a certain family. And you don't mix those two families. Pumps are over here. Uniports are over here. You don't mix the two. Okay? So there's a reason for being restricted. Okay? Now, so that's all diffusion of some type. And let's talk about active. So let's come down here, give ourselves some room. So this is active, and it is mediated by, it's got to be a protein. That's one good rule. Simple, you've got a choice of just diffusing through the membrane. That's a possibility, very limited possibility. Most diffusion is facilitated using proteins. All active transport is using proteins, membrane proteins. What kind of protein? Do you remember? What's it going to look like? Integral group. If you're having to refold and face both sides of the membrane, you better be a transmembrane. You better have it's a multi-pass membrane protein. It's a transport active. All right. So there's a couple of choices. It's mediated by what? Okay. There's primary active. You notice Dr. Ortigas mentioned this, and that's where the pumps come in. These are ATPases. If it's primary, if it's splitting ATP to move a solute, that makes it a primary active mechanism. And we also call these guys, they're a type of transporter, but we also, I will probably call them only pumps. Okay. Now, if you have primary, there must be a secondary, right? Secondary active. And that's where the energy is ultimately derived from ATP hydrolysis, but the proximal source of energy is an ion gradient. Okay? So you have symports, okay? And what that means is if you have an ion gradient, you're using that as an energy source. You're actually moving, you're trying to move something else up against its concentration gradient or electrochemical gradient. That's the solute of interest. The sodium gradient, for example, is set up by the sodium pump, right? And you're going to let sodium come down its electrochemical gradient, and that energy will be captured by the solute that's moving against its electrochemical gradient. That's how glucose gets into your intestinal cells. It's fairly uncommon, but the sodium glucose transport protein is a coupled mechanism of transport. It binds two sodiums and one glucose and goes in the same direction. Hence the term symporter. It's going in the same direction. Simple. So there's always more than one solute. One solute is providing the energy, usually it's sodium, or sometimes it's a pH gradient. The other is the solute that you're trying to move against its concentration, like glucose. And I'll show you an example. I just want to give the broad strokes there. So if you have symport, you must have a right? That's where, let's say, sodium comes in and it kicks out chloride. So that would be a sodium chloride anion. Now, what band 3 is, now we complete that story, that band 3 transporter in the red blood cell membrane is a chloride bicarbonate anion. Chloride bicarbonate. You notice there's no charge moving, right? Because for every bicarbonate -1 that goes in, you have a -1 that's leaving. So it's not electrogenic. It's not generating charge. Now, those of you that went to Dr. Ortigas' lecture, is that electrogenic or not? Does it produce current? So 3 sodiums are going out. How many potassiums are coming in? 2. So it's not equal, right? So it's actually making the plasma membrane more negative.
the inside. So the way you do this, here's a rule. So if you don't want to think about it, just take in minus out. The things coming in, 2 potassium, minus 3 equals minus 1. Right? And then you're doing that on this, you always start at the side of the glass, do that little calculation, and you'll always know whether it's electrogenic or not. And some pumps are moving positive charges, and some are moving negative charges. But whether it's electrogenic is an important topic. Because if it's electrogenic, it's having to move against an electrical gradient and a chemical gradient. Okay? So antiports, imports, and then just to remind you, no uniports. They don't belong here. Okay? Uniports cannot, since they only move one thing and they don't split ATP, they can't be in this category. They're only up here. Now, I noticed I didn't finish one aspect of that. It's channels. We're not going to study channels right away because they're pretty complicated. So we'll get to that when we talk about neurotransmission. Okay? But ion channels, there's different kinds. There's ion channels. There is water channels, which is weird, but they are. And then there are urea channels. And water and urea movement is important in your kidney. That's how you excrete nitrogen. So that's the whole everything we're going to talk about fits into those different categories. Now let's do a real. Let's see how we do on time. Any questions about this? We're going to reinforce it, right? Okay. So we've done this. Come on. We've classified proteins. So now we want to get some real poster children that are developing. And so we'll go through this pretty quickly. One of the things that there's always 3 questions to ask about transport mechanisms, no matter what you're looking at. Okay? The first is the chemical nature of the transported solute. The second is what's the energy that's required to move it? Is it passive or is it active? So that's the second question. And then third, what's the mechanism? Is it simple, no protein, or is there a protein? What family of proteins is it? So those are the 3 questions. Okay? So that's the way you should think about things. And the first question is important because of this diagram right here. Very few things are going to enter the cell by simple diffusion. And that makes sense because the fluid mosaic model means that everything is going to try to make this barrier that's exquisitely impermeable to everything except small things and gases. Everything else is going to be chosen. And so only the good stuff the cell wants comes in. Now, we can fool it with drugs and all these other things, but that's a different idea. So it can be lipophilic, it can be a gas, and it can be small. Those are the only things that have appreciable, simple diffusion through the membrane. And even steroid hormones, we're finding out, they have transporters as well. So it's pretty strange. Okay, so even if you get as big as water is small, right? The reason you need to have a channel for water to move is it's polar, right? There is a finite simple diffusion coefficient for water, so some of it will leak through because those phospholipids are moving around, and every once in a while a water molecule will shoot through there, once a week or something. But you can't depend on that. Your cell would burst before you got enough water, right? So water, urea, glycerol, those are all they have channels to mediate. So most of it can't go through. That's simple diffusion, that's a small fraction. Okay. Then when you get to things that are still small, like glucose, sucrose, sugars, they're polar, they're big. Glucose is about, 181, I think, for its molecular weight. That's way too big. So that's not coming in unless you transport it using protein. And then, so if it gets big, you have to use a transporter. And it gets charged. If it's carrying a full charge, it's never going to go through the plasma membrane by a simple diffusion. It's going to require a channel or a transporter of some nature.
All right, so that's why chemistry is important. The second thing is the energy. So you always ask yourself, is it going down its electrochemical gradient, or is it going against it? So you piece it together this way. So this is charged, okay, and there's only one case. So if it's electrically charged, you have to consider two things. The chemical potential, which is, this is higher on that side, okay, so it's going to move down its chemical potential, which is towards a lower concentration, so it's down its concentration gradient. But if there's no charge across the membrane, you can forget about the charge, even if it's charged, right? So if the plasma membrane, which is unlikely, is zero, if the plasma membrane potential is zero, you can forget about charge, okay? But that's the only time you're allowed to forget about the charge, or that source of energy for a charged particle. Mostly you get this situation, where there's some sort of membrane potential. Now, membrane potential is just a difference in charge separation between the inside and the outside of the cell. And so if we stick, so Dr. Ortigas was talking about putting electrodes in a cell. So if we put an electrode in cytoplasm, usually cytoplasm is down, okay, the convention is outside the cell. If we put a electrode here, and then connect it to a voltmeter, and then put the ground outside, the outside of the cell is always taken to be ground potential. What does that ground mean? I mean, it's literally, you take that electrode and you connect it to something that goes into the ground. I mean, that's serious. The Earth is zero potential, okay? So outside of the cell, by convention, is taken to be zero. The difference is what's going on inside the cell, and a typical Joe Blow membrane potential to memorize for, if nobody tells you anything else, it's going to be minus 60 millivolts. And when I say minus 60, with respect to what? Outside the cell is zero. Inside the cell is minus 60. So that means there's a lot more negative things on the inside than there are positive things, okay? And that's the situation, this is the classic situation, okay? And so if you know that there's a membrane potential, you don't even need to know the magnitude. If there's charge separation, that's going to be an energy source. And if you look at your solute, and if it's charged, you have to think about the membrane potential. Okay? So if it's positive, and this is just common sense, if it's positive, it's going to move this way based on the membrane potential, right? Which way is it going to be moving based on the chemical potential? Inward. Okay, so you don't need any fancy equations. I'm going to give you one, but you don't need it to figure this out. This is common sense. The chemical potential is directed inwards. The electrical potential is directed inwards. You don't need any math. It's going in. If what happens? You have to provide a pathway, which is the protein has to open and close. Okay. So that's the third part. You know, once you consider the nature of the solute, the driving forces, this can move based on diffusion, because it'll come in based on its energy, the energy separation. The next way is, how do you get it across an impermeable membrane? You put a protein in there, and that's the mechanism part of that equation. This is where it gets complicated, okay? So this is where you have to have some mathematical technique to figure this out, because the electrical potential is opposing the movement of this. Let's say it's sodium. Sodium is a nice example. It's got one positive charge, right? When does a cell become positive on the inside? Can you think of a cell type that sometime in its life becomes positive?
During the action potential.
In a neuron. So all of you, I hope, are producing lots of action potentials moving down your neurons at the moment. What's happening is the inside of the cell, before that action potential gets there, is negative. It's probably closer, about minus 80. And then when your thought process goes through there, it depolarizes and becomes positive on the inside. So you tell me which way sodium is going to move under these conditions. Chemical potential is higher outside than it is inside. The electrical potential is opposing it. You can't tell me without some math. You have to know the concentration on either side. And I'm going to give you that tool. It's real simple math, but it's called the Nernst equation. And I'll teach you how to use it. But that's how you tell, is you ask the question,
you have to ask the question, what's the concentration? And if you know the membrane potential, you can calculate it. All right. Okay, so let's quickly go through. So these are the cartoons of these various things. Simple, sorry, Dr. Artigas had the same problem that I do, so it's not just my fat fingers. Simple diffusion, no protein. Okay. Channel, it's a multi-pass membrane protein. We're going to talk about it later, but they're all multi-pass. Open and close, open and close. Okay. This, and then transporters, they move much slower. This is the fastest mediated transport at about 10 to the 7th, 10 to the 9th ions per second. Really fast. Transporters, the next fastest, it's about 1,000 to about 10,000 per second, so it's slower. Okay? That's because the solute has to bind, it has to refold, and then it has to face the other side, and then it comes out, so it's slower. Okay? And then active transport is where you've got some sort of energy input. If it's ATP hydrolysis, it's called primary active. It's coupled to a gradient, so that's two solutes moving, and one of them is piggybacking on the other, that's secondary active. Okay. You'll get examples. Okay. This is just sort of a close-up look. What kind of protein is this? Can you tell? In that category. If you look at your notes and look at this is protein. So look at the concentration gradient. Okay. So it is going down its chemical potential. There's no charge. They don't say anything. But even if this is charged, it doesn't matter because there's no charge across the membrane. They didn't give that to you. So you can assume it's zero. So it's a concentration driven, so it's some sort of diffusion. And so if it's some sort of diffusion and it's a protein, there's only two possibilities, a channel or a uniport. Okay. Now, this is what's confusing to students sometimes. They go, well, it can be uniport, there's two things going through this. No, it's not the stoichiometry, it's the type of stuff that's going. This is only one type of molecule. It doesn't matter if each transport cycle carries two, but if it's two sodiums, that's still just a single solute, that's a uniport. If it's two different solutes, that's different. That's going to be coupled, transport, secondary active. So, again, the stoichiometry, that is how many of those things move every transport cycle, is unimportant as long as it's just one thing that's moving, one type of solute. So that's a source of confusion. Okay, and then channels, we'll get to that. They don't refold. What they do is there's a gate that opens and closes. So we have to talk about the molecular nature of gates. Okay, this is just coupled transport. Coupled means there's more than one solute that their movements are required to go together, either in the same direction, symport, or the opposite direction, but the movements are coupled together. One cannot move without the other. That's what coupled is. That's secondary. And then if the thing is splitting ATP, it's a pump, or we also call it an ATP. And that's what Dr. Artigas was talking about. But you can use other types of energy. We don't use this in most eukaryotes, except the plants do that. But that Bacteroidopsis can actually use light as a source of energy. It traps a photon and it causes a proton to be ejected. Or you can use the electrochemical gradient as a source of energy. And these are the definitions for uniform symmetrical gating. We'll come back to that. Now, one of the properties that I need to point out is the speed of the transport and what does it look like. So the kinetics of the transport inform you of how it's moving across. Okay? If it's a straight line, it's simple diffusion. And all concentrates, so you're plotting, so this is like a Michaelis-Menten plot. Except this is, it's not a reactant as you would plot here for Michaelis-Menten. It's a solute concentration.
So this could be glucose. Versus what? Well, it's the initial rate. It is like Michaelis method. In fact, you don't have to learn any new fancy kinetics. Most of these transporters follow Michaelis method kinetics. So Km means the same thing here, almost, that it did in enzyme kinetics. So good, we don't have to learn anything new. There's a Km. What does that tell you about the protein's binding affinity for that solute? If it's a small Km, it's high affinity. If it's a sloppy Km, it's large, it's a sloppy affinity. So it's inversely related to the affinity, the Km. There's a Vmax. What does that tell you? It tells you about the number of transporters and the speed with which each one does its job. How fast can that one monkey move things across the board? And then times the number of monkeys. That gives you the Vmax for the transport. Notice, if it's moved by a protein, it's a square hyperbola. It maxes out. So it will it's called saturated. It's saturated. Diffusion is not saturated. So from this, it's square hyperbola. It's faster than simple diffusion, and it's saturated. Those are the characteristics from its kinetics. Any questions about that? If we didn't have to learn anything, we would do this. Okay, let's begin, and we're only going to get this one. We won't even finish it. Let's get some poster children. This is an entire topic. Facilitated diffusion. What uses that? Organic nutrients. That's how you get all your energy from your intestinal epithelia. You're moving sugars, amino acids, nucleotides, phosphates, all kinds of things. And they're going to be using, they're going to be using a couple of different types of transporters, but we're going to focus in on a class that moves glucose. Okay, that's going to be our poster children for this facilitated diffusion. And we know what the energy is like. Glucose cannot go into most cells unless the concentration of glucose is lower inside the cell than it is outside. So it's driven by the chemical contention for most cells. There's a couple of exceptions. And so it's a uniport, and the family is called the GLUT family. And so what that stands for is glucose transporter, GLUT. Okay. And it's just GLUT 1, 2, 3. There's actually 12 family members. What kind of protein do you think it's going to be? You should just immediately say it's a integral, multi-pass membrane protein. This family has 12 membrane-spanning alpha helices. The N-terminus is on the inside of the cell. Where's the C-terminus? It's the real even, same side, odd, opposite side. So the N-terminus is on the inside, the C-terminus is on the inside, both sides. Okay, this family is really good at distinguishing between glucose and anything else. So these are two sugars that are highly related, mannose and glucose. This number is going to tell you what the affinity is of that transport for that particular solid. Look what happens. So for glucose, the Km is 1.5 millimolar. All you do, if you take this hydroxyl in this chair position, you flip it up top, so you got uh-oh, and then oh, over here, that changes the whole setup. Look at the Km. So the solvent of the solute that a transporter is using is going to affect the Km that you measure. That's a really important take-home message is the Km can be affected by the solute that's being transferred. It doesn't want this, so it wants glucose to come in. Now, what's interesting is you've got different family members of this protein. There's different family members in different types of tissues. And so GLUT1 is in your red blood cell. It was called that because that was the first one that was identified. And there's GLUT2, that's in your intestine, facing the blood. Okay? GLUT3 is in your brain, that's what keeps you thinking. Okay? This one has really high affinity. That's because the brain is the prima donna. If you deprive the brain of glucose, what happens to you? Okay, so it gets its cut right off the top. Okay. GLUT4, that's the insulin-dependent one. That's what's controlled by insulin. And that insulin receptor is in muscle and fat, so that's what that's where this transporter lives, is in your muscle tissue and your fat tissue. GLUT5, what's this mean? The Km is infinite. So this thing will not transport glucose. It's the fructose transport. So all that high fructose stuff you drink and eat, this is the one that takes it in, not. It's in the same family because it's got the same structure and smell and everything, but it's the fructose transporter, it's GLUT5. And we'll use that to move some of your nutrients when we get to it. But look at the values. There's something that makes sense here. The highest value is the sloppiest affinity. Which one of these things do you put, what tissue is going to encounter the highest levels of glucose? You don't breathe glucose in, do you? No, you eat it or you drink it. So you put the one that's got the worst affinity there in your gut, okay? So this is the slop. It doesn't need a high affinity because you're going to have lots of sugar coming in, okay? Where does it go next in your body? It goes into the bloodstream, right? So this one is the next worst affinity. And this is the worst. This is 7. So it's going. So the blood can suck it out of the gut. Okay. Where does it go next? Which one's next lowest? The GLUT4. The one that's. This is the insulin is going to store the glucose either in your muscle tissue. The major consumer of glucose that you eat is muscle tissue. That's because the insulin just packs it in there. And then after that's stuff, then it starts to turn it into fat. So we're going from gut to blood to fat, and this guy has the highest affinity for glucose so that it can take its cut right off the top. The brain always demands its own cut. So the highest affinity is in the brain.